In the last unit, we learned about how these different types of elements, such as carbon, oxygen, nitrogen, would come together to form various compounds. And the various compounds could be known as, lipids, nucleic acids, such as DNA and RNA, proteins, as well as carbohydrates. What is really unique is that how did all of these organic molecules eventually form cells? So that's a huge question. From these organic molecules and ultimately forming cells, huge amount of time in between. There wasn't anybody to really be around at that time to give us some sort of eye view or any sort of documentation. They couldn't take pictures. There's nothing there because we're looking at about 3.5 billion years ago. And you think about that, write that out in how many zeros, and that's a long time ago. So what we have to look at now are different things such as evidence, chemical evidence, current biological evidence, to somehow make the connection on how these organic molecules ultimately formed cells. We can consider this a biological evolution. In other words, a huge leap. In terms of the whole concept of evolution, one of the most accepted one is called punctuated equilibrium, where there are long periods where nothing really changes and then something causes huge amounts of change, remains stable for long periods of time, and then change occurs again. Something very similar to what Darwin noted in his travels, but we're looking at something that's very different than what Darwin basically is going to be considered how unique organisms are in terms of linked to each other from different points of fossil evidences, but we're looking at something totally different. We're looking at the biological evolution. We're looking at a biological evolution in which these organic molecules, non-living, ultimately coalesce to form something living. Unique was the Earth's atmosphere. It has been the same since. It has been tried to be replicated in various incidences such as Miller and Urey, but we have to recognize that today, in terms of 21% oxygen, very different than what it was 3.5 billion years ago. It's much lower. Who knows why? It could be the fact that more aerobic respiration has occurred. Could be a stabilization. We also know that in terms of some of the different gases, they also are in different balances. So what the atmosphere was 3.5 billion years ago is very different than what it is today. So we will not see this type of biological evolution in our lifetime. We do know that in order to have some type of cell, we need organic matter. The organism, the cell, has to be able to replicate. Precursor to DNA was that of RNA. DNA is a more stable compound, being double-sided. A cell also has to have some sort of mechanism of metabolism and the ability to use ATP to do work. But most importantly, it has to have a plasma membrane. The plasma membrane is going to be significant because within this membrane, the membrane can basically form a boundary between what's outside of the cell and what's inside of the cell. And inside of the cell, reactions such as metabolism, replication can occur that could be different than what's outside of the cell. So the plasma membrane is going to be key in terms of the integrity, or what we're going to see, the homeostasis that all cells contain. And by being able to remain constant inside by regulating what goes into or out of the cell, it can remain different than what's on the outside. One person, Lynn Margulis, if you go ahead and Google her, I went ahead and here's the site here. She basically started this whole concept of what we call endosymbiotic theory. In other words, endo, inside, living together. Her whole premise was that in conjunction with these things of the small steps in evolution, that there are organisms that ultimately joined, such as a spirochete bacteria with a single cell bacteria, Or perhaps another concept that she came about would be prokaryotic cell. Prokaryotic cell. One engulfed the other, so now we have one cell living inside another. And so therefore, this is the premise of her particular theory in terms of what we call endosymbiotic theory. We also know that there's an endomembrane, and there is also the correlation between what these organelles such as mitochondria and chloroplast have to help support her particular theory and writings. When you think about here, there's two evidences here about these protobacteria and the cyanobacterium. Cyanobacterium is very closely related to the chloroplast in terms of its structure. How do we know? The DNA sequence is very similar. Then we have here the mitochondrial DNA as well. So in other words, mitochondria and chloroplasts have their own unique DNA. These particular organelles are able to replicate under binary fission independent of its host cell, or what we might consider its eukaryotic host. Notice that these particular events came about 2.7 billion years ago, after a lot of the organic molecules had already established themselves as being stable. Here's another depiction in terms of how it's possible to support this oxygen-breathing bacteria and the photosynthetic one. These essentially diverge to form what we call animal cells and plant cells. It's very possible that the membrane began to enfold in on itself, and that's how we began to get these organelles called the endoplasmic reticulum, as well as within here, nucleic acids, more specifically DNA, became enfolded and enclosed within a nuclear envelope. So in other words, now we have the nuclear material that is unique from the cytoplasm, and at the same time we have the mitochondria and the chloroplast unique within the actual cell itself. So here this endosymbiosis is starting to kind of give hold in terms of more and more evidence to support Margulis' theory. What we know, when we look at the evidence, we look at the sequence here. We look at the sequence of the DNA, we look at the sequence in terms of the RNA, in terms of the nucleic acids, mitochondria and chloroplast. We're going to compare that with bacteria. And when we find that comparisons, we find that similarities are too unique to think that they were anything different. So this is what we're looking at in terms of evolutionary biologists, or we may be looking at more specifically in terms of evolutionary microbiologist, because we're looking at a very small scale. Remember, Darwin, different. Darwin saw the macro, the big picture in terms of the fossil evidences and structural, the homologous structures, analogous structures. We're looking at very, very small structures that you need today's modern technology and sequencing in order to establish. Thus, if you were to read a biology book 15, 20, 30 years ago, it would not have the same type of evidence as we have now, simply because as technology has increased, so has our understanding. The other thing we're going to see is this 922 pattern here. We're going to see the basal bodies. This basically is going to be the hook or a unique structure that is going to be common in things such as centrioles, which are found in animal cells. This is also going to be a structure that is going to be very, very similar in flagellum, as well as also the tails of sperm. So this 922 pattern is unique in many different organisms. Thus, it gives us yet another piece of evidence to support this endosymbiotic theory. It's very possible that these particular structures are very similar to what we call the spirochete bacteria. I know I misspelled that. Spirochete bacteria are basically these corkscrew-like bacteria. They can cause diseases such as syphilis, elephantitis, etc., but they also have a very similar pattern to these eukaryotic organism structures. One of the things we want to look at is how these cells, the eukaryotic cells and prokaryotic cells, how are they similar? What's unique to both? What is similar to both? And then what is going to be common to all eukaryotic cells? Eukaryotic cells being those in the kingdom Animalia, the kingdom Plantae, the kingdom Fungi, and the kingdom Protista. Prokaryotic cells are going to have two domains. They're going to have the archaea and the eubacteria. So what we want to see is what's going to be similar to both types of cells and what will be different. And that's for you to kind of divulge into and figure that out. Maybe go ahead and make a diagram yourself, write it out. How are they similar? How are they different? How might you support this concept and theory of endosymbiosis?